So let me invite all the speakers up to the stage, and we'll take questions from the audience. I think while people are coming up, I think we've spent a lot of time the last couple days talking about drug development pathways and biomarkers. We can continue those discussions, or we can talk about specific programs depending on what your interests are. If you have a question for a specific presenter, please address it to that presenter. Otherwise, if you address a question to the panel, we'll ask for volunteers to answer it. So if we're not all talked out from the last couple days, let me invite anyone to the microphone who'd like to ask a question. Okay, I think, so oh, Andy.
Why not? I did have a question for Charles. You mentioned 2 water dispersible formulations, I think, and I was trying to clarify which is which and what were the target populations for those. There was a P100H100 and a P150H100.
Yeah, the P100, it's each individual, the rifapentine water dispersible and the isoniazid water dispersible separately. It was a little confusing on the slide, and then the segue into the fixed dose combination that's water dispersible.
It seems like a quiet group. I'll throw one out there. So I'm Joshua from Thermo Fisher Scientific. I'm curious, as new drugs are actually developed, and obviously there's a growing pipeline, which is fantastic. When you think about this and you look at the diagnostics component to this, and you think about the resistance and the speed in which resistance develops, I don't know if this is a question you can answer, but how do you think about that with the new drugs and the availabilities into the regions where it's most applicable, into Africa or Russia or China, India? When you think about these places, how do you think about resistance? Do you think about it from a molecular perspective? Is it important to include in the diagnostics, or you're like, oh, those drugs are going to take quite some time before they ever actually get it to the markets where they'll be deployed? Or do you look to someplace like Chris, WHO, or the TB Alliance to try and integrate that? Like, what's your viewpoint from a drug development side of things and integrating that? So, thank you.
Connie, do you want to answer that in terms of what we do? Because that's one thing that I didn't include in the update. But
one comment I can provide is the fact that in terms of new drug, bedaquiline, for example, we're trying to incorporate that into existing platform in terms of diagnostic, for example, the sensitive titer. But it has to be understood that there are no breakpoint at this point for those new drugs, so interpretation is not there, but we are collecting data to generate enough data for the regulators to make breakpoint. But certainly it's being looked at in the case of bedaquiline.
I can add, we have a number of our diagnostics colleagues in the room. Yonka, do you want to add anything about how resistance testing has been incorporated within the delaminated program? I know we've had some early collaborations in the last couple years, and
right. I think from our side is we always cautious on when you implement including delaminate into the treatment, because we want to make sure that you monitor acquired resistance, emerging of resistance. So we have been helping with request of outside government or hospital that if they want to delaminate was provide delaminate to them under agreement, so they can do measurements of resistance emergency monitor the resistance development. So that's usually we do, and also we're helping with other consortiums to develop the DST, immediate-based DST, or Thermo Fisher. We're helping with their to develop the 96-well plate MIC plate to to implement DST measurement in the field.
And as Charles just mentioned, working closely with EMA early on to develop a breakpoint.
Thank you.
So maybe one aspect to diagnostics and resistance might also be to look for molecular markers of drug metabolism to identify patients at risk of having really low drug concentrations and thereby being at risk of acquiring resistance. And we don't, for the drugs that we have, the new drugs that we are looking at in Panacea, we don't seem to have markers for that, but we're currently looking at whether this is something we can do for rifampicin. But yeah, see as time passes.
Thank you. ID?
Right. ID Rusin from the Union, and we're the sponsor of the STREAM trial. I know this could be for anybody. It's very promising that people have lots of plans for future trials, but just a reminder of site development, and the trials have to take place somewhere, and especially in terms of MDR-TB trials, I think that that is an important area not to forget. Thanks.
Hi, everybody. I have a kind of question for all of you who represent really different sectors and have products at different stages of development. I wanted to follow up on Wim's earlier question about compassionate use, which was directed towards the TB Alliance, but I think it's a good entry point into something that advocacy community activists have been thinking about, which is having some kind of overarching compassionate use structure where it's not either the responsibility or the privilege of individual companies to determine who get the drugs and who don't, and would potentially maybe even allow for compassionate use of products used in combination when potential benefits would outweigh the risks. And I'd love to hear thoughts from all of you guys who are involved in the actual product and regimen development about whether that would be something of interest. You know, it would mean that you might not have to do as much work to find the resources to make it happen, and that could be, you know, outsourced or at least pooled. But also it might mean relinquishing some of the control. On the other side, then you also potentially would, you know, limit some of the liability, which I think has been a concern for some of the developers. So I'd love to hear, you know, from the various sponsors who, like Janssen, started a compassionate use program quite early and has a lot of experience, to Otsuka, which, you know, got started a bit later and has more limited experience, to TB Alliance, which hasn't been able to get one off the ground yet because they rely on outside funding as a PDP, as well as some of the groups involved in much earlier stage product development.
Yeah, let me pass this down to the two that have a program in place. My understanding, one challenge is that so much of the mechanism of what one has to do is really country by country by country specific. The legal structure in different countries.
Right, so that would be part of the idea, is like if there was a central body coordinating it, they would know the deal in each country, and each sponsor wouldn't have to reinvent that wheel every time they want to make compassionate use available.
I mean, we're willing to share our intelligence if that were to help. But it's interesting that you brought this up now, because when we started compassionate use, we actually were looking to put together a, how did we call it, a sort of a committee, advisory committee, I think we called it, to actually review the cases, so that we wouldn't make the decision, but we would have experts in the field making the decision. Nobody was interested to participate in that. So we started by ourselves. What we do, so our compassionate use has now been completed, but we have a lot of experience from our HIV compounds, so that's where we drew the intelligence from. We do have a compassionate use program open for pediatrics now, and for that we do work with a consultant who can give us advice on the cases that we're getting in. So that's where we are.
Hi, Erica. A couple of thoughts about your question. So I can say that there have been partnerships going back a couple years since I've been working in compassionate use at Otsuka. Initially, we worked very closely with MSF. Now that they've grown their NTB program, that's been less of a project, but we work very closely with European Respiratory Society, TB Concilium. So in a sense, that is an available mechanism for any developer of drugs to work with, because they have an ongoing site and system where they will receive cases and review it independently of the company and determine whether they think a case does merit compassionate use or not. It is still an issue that every country has its own regulatory framework with regards to compassionate use. So there are just some countries that I understand will not do compassionate use, period. So I don't know what the workaround is in those sites. Obviously, the developers, producers have to be on board with it as well. There's cost issues, which you all raised. I don't know, Charles, do you have any?
Safety surveillance. I mean, just that's the other part of what we discuss a lot.
Yeah, so that Charles brings up a really important point. I'm sort of more on the clinical end, but perhaps for developing a new drug, once you send it out for compassionate use, what sort of pharmacovigilance monitoring is in place? How will serious adverse events be reported and recorded? Who will receive those cases? How will those be reported to the local countries and other regulatory bodies? So those are some of the initial thoughts I had.
So I must admit I don't have any experience in that field, and being from a rather small academic group, hearing about the complexities, I think your idea about setting up a framework that can be used for multiple agents sounds attractive, and we might spend some thought on whether this might blend into the real-world regimen development pathway in any way in the long run.
Yeah, we could encourage any funders who would like to help set up some framework like that. That would be wonderful.
I think just wanted to add on that, obviously from MSF working in developing systems to access compassionate use. We started discussing with Janssen a couple of years back. I think it's very clear that it's a heavy process, and you need the right expertise to be able to also push back to manufacturer to say, no, these patients actually need this. Don't be too conservative on this. So I'm just thinking that there are obviously kind of like now from in the European region, the kind of consiliums, or where difficult MDR-TB or XDR-TB patient information can be shared and advice given. Perhaps those are the kind of places that there can be some suggestions if they can take on compassionate use process. But as I say, from our experience, it's a really, really heavy process, and relying on people who are just volunteering it's quite difficult to do.
Thank you.
We're 5 minutes over time, so okay. So we'll, thank you. Aminu, just very quickly then.
Can I just put it to you all that we already have a highly effective regimen for drug-susceptible tuberculosis, cures 95% of the patients. The problem is that the persisters who then cause a relapse in the remaining 5%. And I've just started a new trial, the REFRESHOR trial, in which we'll be looking at this population of persisters using RPF and transcriptomic studies. I'd like to know, are you building in any such studies in your drug development program?
I think that's a very good question, and also with respect to the data that comes from Jeremy Coates' group. Certainly we don't have something as elaborate as what you described, but what we are trying to do is to look at molecular bacterial load assay data, which we would hope captures persisters as well as multiplying organisms, but that's still underway. No results yet.
Okay, well, thank you. I'd like to thank the presenters. And the audience for your participation. Thank you.